Biology Part 9. The structure material of insect exoskeletons is carbohydrate derivative called chitin. Like cellulose, this is a polymer of the linked subunits, but the subunits themselves differ from glucose, having one of their OH group replaced by a more complex NHCOCH3 group. This difference means that there is even more hydrogen bonding between chains than in cellulose, making chitin extremely tough and resilient. There are chemical tests for carbohydrates. The common chemical tests used to distinguish different carbohydrates are as, say for instance, there is Benedict solutions test, Fehling solutions 1 and 2, Benedict solution, iodine in K1 solution, iodine in K1 solution, Schultz solution, acidified ferroglycine solution. Next we talk about the structure of proteins. Well, first of all we need to understand the types of proteins and their functions. The proteins make up about half the carbon compounds living organisms. Although they share many basic structural features, their overall organization is extremely flexible so that individual problems can have very different and highly specified functions. All proteins are polymers made up of subunits called amino acids, joined together in long chains which may be folded or twisted in various ways. Two main groups of proteins can be distinguished according to the high chains which are arranged. In fibrous proteins, the chains are extended so that the molecules are long and thread-like. Partly as a result of fibrous proteins tend to be insoluble and comparatively resistant to changes in temperature and pH. The properties make the fibrous protein ideal as structural materials. Important examples are collagen and elastin, major proteins in skin and connective tissues. The keratin in hair and hose, nails and wool. The second major group of proteins are the globular proteins, in which the chains are folded in more complex way to produce compact mounded molecules. These dissolve easily to form colloidal solutions and are often susceptible to small changes in temperature and pH. Globular proteins are functional proteins of metabolism. In other words, they carry out many specialized tasks which enable the organisms as a whole to function properly. Many of the globular proteins are enzymes that are specific catalysts and altering the rates of most of the chemical reactions of the body. More than 2,000 enzymes have been isolated in pure form, and they are particularly important because, as will be discussed in next part, they provide normal means whereby the genetic instructions coded in the nucleus of a cell are carried out. Well, the globular proteins also act as hormones, antibodies, blood transport pigments, light sensitive pigments, and blood clotting agents. Some of these proteins and many enzymes as well cannot function unless joined to a non-protein component called prosthetic group. Such proteins are called conjugated proteins, and they include hemoglobin, the pigment in red blood cells. Hemoglobin molecules are able to transport oxygen because they contain prosthetic groups called heme group, and which are the integral part of their structure.